Good morning, grade 10. How are you? Today, inshallah, we will start with chapter 13, lesson 4, gene regulation. This lesson will take 2 videos, inshallah. Let's start with the outcomes. We need to identify the prokaryotic gene regulation in this video, and the next video, inshallah, we will identify the eukaryotic gene regulation. In the previous lecture, we studied how we convert the DNA into RNA and how we will convert the RNA into protein in a process called transcription and translation. And inshallah, today we will study the gene regulation. How are prokaryotic genes regulated? How we can regulate the prokaryotic gene? You know, the prokaryotic genes are simple, small, so it differ from the eukaryotic genes. DNA binding proteins in prokaryotes regulate genes by controlling transcription. So we have a DNA binding proteins in prokaryotes which regulate and control the transcription. Let's start with the bacterial DNA, because bacteria is one of the prokaryotes which do not need to transcribe all of their genes at the same time. To conserve energy and resources, prokaryotes regulate their activity, producing only those genes necessary for the cell to function. For example, it would be wasteful for the bacterium to produce enzymes that are needed to make a molecule that is readily available from its environment. So the bacteria will not transcribe all the proteins or all of the genes at the same time. By regulating gene expression, bacteria can respond to changes in their environment, the presence or absence of a nutrient as an example. What makes it important, which is the DNA binding proteins in prokaryotes, regulate genes by controlling transcription. Some of these regulatory proteins help switch genes on, while other turns another genes off. So the genes in bacteria are organized into operons, and operon, which is a group of genes that are regulated together. So all of the bacterial genes are regulated into or organized into operon, and operon, it means a group of genes that's regulated together. The genes in an operon usually have related functions. So the genes in an operon usually have related functions. Like an example, we have the 488 gene that code for proteins, 4,288 genes that code for proteins in E. coli, include a cluster of 3 genes that must be turned on together before the bacterium can use the sugar lactose as a food. So we will study the lactose lac operon. So these 3 lactose genes in E. coli are called the lac operon. So we will study the lactose gene, 3 lactose genes together. The lactose is compound made up of 2 simple sugars, galactose and glucose. To use lactose for food, the bacterium must transport lactose across its cell membrane and then break the bond between glucose and galactose. These tasks are performed by protein coded by the genes of the lac operon. Okay? If the bacterium grows in the medium where lactose is the only food source, it must transcribe these genes and produce this protein. If it grows on another food source, such as glucose, it would have no need for this protein. So the lac genes are turned off by proteins that bind to DNA and block transcription. So again, lactose is made up of 2 sugars, galactose and glucose. And at the same time, if the bacterium grows in a medium where glucose or galactose is found, then there is no need for the lactose genes or lac genes. If there is no food resource except lactose, so we need to switch on the lac operon or the lac gene. On one side of the operon, 3 genes are 2 regulatory regions. Okay, the first is a promoter. So this is the promoter, this is the operator, and we have before it the lac repressor gene. So we have 3 locations. So the first is a promoter B, which is site where the RNA polymerase can bind to begin transcription. The other region is called operator, which is where the DNA binding protein known as a lac repressor can bind to DNA. So this is the lac repressor, promoter, and operator. Okay? The lac repressor gene codes for repressor protein, which will bind to it. Promoter, RNA polymerase, will bind to this site, to the promoter. Operator, which contain the repressor binds sites, okay? Which is the lac, 3 lac genes. The lac repressor block transcription. When lactose is not present, the lac repressor binds to the O origin, okay? Or O region, blocking the RNA polymerase from reaching the lac genes to begin transcription. So again, when lactose is not here, there is a lac repressor which will bind to the O region, operator region. Okay, once it's attached, it will prevent the RNA polymerase from binding to the promoter. The lac repressor protein has a binding site for lactose. Okay, when there is no lactose, when lactose is present attached to the lac repressor and changes the shape of the repressor proteins in a way that causes it to fall within an operator. When there is no lactose, the RNA polymerase will bind to the promoter and to transcribe the DNA to form the mRNA, and that's it. Once there is a lactose attached to the repressor, then it will remove it, and the promoter will, the RNA polymerase will attach to the promoter to end the transcription process. With the repressor no longer bound to the O site, so the repressor now it's not binding to the O region, RNA polymerase can bind to the promoter and transcribe the gene of the operon, and in the presence of lactose, the operon is automatically switched on. Once we have a lactose, this is small region, it will attach to the repressor, and that will open the O site, and the promoter will attach to the RNA polymerase to finish the transcription. And that's all about the gene regulation in the prokaryotic cells. Have a nice day, and thank you all. Thank you.